My name is Patrik Sundh. I'm the CEO of Cline Scientific, and I will tell you where our company is today. Cline has, according to our plan, transformed from a platform technology company to a full-fledged biotech company with in-house projects that I will tell you about. We have two main R&D projects, one that involves cancer diagnostics and another that involves stem cell therapy for repair of damaged cartilage. We use our patented nanosurface technology by which we can study and control how live cells respond. These aspects are of great importance in accurate measurement of metastatic potential in cancer, as well as the development of stem cell therapies. We have previously presented our patented gradient technology, so I won't spend too much time on that, but we have now embedded this in these two projects that I will talk about. But first, the Cline business model has come in four different stages. The first one meant that we developed and patented our nanotechnology. The second one, we sold products from that for commercial applications, that is research applications. Those things we have already done. The third stage, which we're now embarking on, is to have our own or co-owned life science project that we develop ourselves, to be able to, in a few years, go into the fourth stage, which is to divest those projects to larger companies or larger partners. Today we have, as I said, two projects. One is called CellRace, which has to do with breast cancer diagnostics. I will get back to that. Another one is called StemCart, which is a cartilage repair therapy project, and I will get to that shortly too. We also have our patented technologies. They're ready. We have cell and production labs at AstraZeneca BioVenture Hub. We have an experienced team of employees as well as advisors, and we have initiated contacts with investors and potentially larger partners for the future. So, to get into our projects, the StemCart is a project aimed at developing a stem cell therapy to repair damaged cartilage in knees and hip joints primarily, although it could be other joints as well. It's a project where we cooperate with researchers from the University of Gothenburg and has done so for a while. Damaged cartilage leads to chronic pain, need for pain medication, and it severely limits your lifestyle. I think people who have this problem knows this. For them, it's a painful existence, and to get rid of the pain is only a dream. And StemCart is a project that aims at realizing that dream for them. The problem is that osteoarthritis or trauma causes cartilage damage, and cartilage cannot heal by itself. You're left to complicated surgery with long rehab times, and it's also very costly, unless you want to live with constant pain, that is. Our solution is aimed at developing a stem cell therapy which is less invasive, comes from iPS cells, induced pluripotent stem cells. It will be available for more patients. You can treat a patient much earlier, and the intention is to have much less rehab time and therefore a higher quality of life. What do we do? Well, here is a couple of pictures from, to the left, chondrocytes from our iPS cells that are very similar or identical to the natural chondrocytes or cartilage cells that are present in the body. Our approach is unique because we use our surfaces to grow the stem cells on where you have immobilized biomolecules that control the growth or the maturation of the stem cells. The level of accuracy that we can attain in this maturation process by our nanotechnology or nanosurfaces is unique, And our method to grow sustainable cartilage is also unique, and therefore we have sent in a patent application for this method. The roadmap forward is pretty clear to us. We have already embarked on it, as I said, for this year. We have patent application, we have further studies, we have publications coming out, etc. And for the coming 5 years, we have a fairly clear path as well that is supposed to lead to the divestment of the project to a larger partner, either through a license or through an acquisition. The market for these type of problems is enormous. There are more than 230 million patients suffering from this each year. The surgery market in the US is huge, and we believe with this type of therapy you can address at least 90% of the total market of these problems. That's our stem cell project. Now to the cancer diagnostic products, which goes under the name of CellRace. This comes from a company that we acquired at the end of last year called Live Diagnostics, and it aims to develop a diagnostic product to actually measure if cells from a primary breast tumor will spread and run the risk of metastasizing throughout the body. Breast cancer is the most common cancer form today. It leads to a number of things: psychological trauma, harsh treatments, obviously constant worry for these patients. And CellRace is a project with aims to save more women from deaths that occur from breast cancer. Problem here is that you have 2 million new diagnoses of breast cancer, new patients each year around the globe. 90% of deaths that occur from cancer from the breast cancer is due to metastasis. In other words, spreading of cancer that is discovered too late. The thing is, though, that of those 2 million cases, only 5% are diagnosed to have a metastasizing cancer at the onset, at the first diagnosis. But up to 30% of all cases lead to metastasis. In other words, cancer spreads. So there is a discrepancy of 25 to up to 25% of these patients that are missed. And also knowing that metastasizing cancer has a very high mortality rate, the 5-year survival rate is only 22%, you can understand that this is a big problem. CellRace aims to help to save lives by early and accurate risk diagnosis, and therefore provide a tool to the oncologist to optimize the therapy for each patient, thereby eliminating risk for under or over treatment. These are problems today, very big problems, and obviously also minimize the suffering from the side effects of these therapies that are very, very harsh today. This is a picture of it's a schematic picture of a breast tumor. The orange marked cells here are in the primary tumor called proliferating cells, but they are non-invasive. In other words, they grow, they become more, the cancer grows, but they don't spread. Those types of cells are easily identified. There are good diagnostic methods, and there are also very good ways of treating this. The problem in breast cancer, though, are what's marked in blue. Those are cells that are invasive, they can spread, and they actually are transformed from the proliferating, the orange cells. The problem there is that there are no good ways of discovering these cells and therefore to treat them in time before they get out in the bloodstream and are spread throughout the body. How is the CellRace product going to be used? Well, a biopsy is always taken from each patient at least once. We get part of that material, we isolate the tumor cells, we add them to our product, our slide, and measure how or if these cells move. Then we collect all that data, we analyze it, and the intention is to give a result that can show the oncologists the risk for metastasis, or if there exist risks at all. What do we do though? I said that we measure if they move. Well, all metastasizing tumor cells have one thing in common, which is the ability to move in one direction, that is out of the original tumor. That is called migrational direction, or directional migration. This is what we, with very high accuracy on our surfaces, can measure, as opposed to the indirect measurements that are available today. We use live cells, so we know what happens to the actual cells directly when they are taken from the tumor. We also measure some other motility aspects, but I will not get into those here. We have some very promising results. We have used cell lines of tumor cells. The green bars are cell lines that are non-metastatic, in other words, they're non-invasive, do not spread. And the red bars that you see here comes from tumor cells that do metastasize, that do spread. And we can, even though the differences are very small, we can very accurately distinguish between them with very high precision. This project also has a roadmap that is very clear. This year we have started our first test on fresh patient material, that is biopsy materials. We have a patent application in the works, and a number of regulatory documentation issues that we need to start, that we have started. And within 3 years we have, the plan is to have a partner that we can either divest it to, or at least to start working with to bring this to market. The cell-based market is, as you know, 2+ million new biopsies yearly. Cancer diagnosis products is a vast market already today, and the potential in just the EU and the US is much more than what is normally called a blockbuster volume for a pharmaceutical. We have a number of people involved, our board of course. We have everyone working at the company, which are 5 people, but we also have a number of collaborations with researchers that do part of the work. The 3 people on the right-hand side are involved in cell race. On the top 2 on the left are involved in stem cell, and we also have clinicians involved that I haven't brought up here, and we will add more. So that's how we can have 2 projects running at the same time. We are in the middle of a new rights issue, as I'm sure people have seen. Our subscription period ends May 28th, or runs through May 28th, and there is also a warrant attached to this that will vest in beginning of March next year. Capital goes to these 2 projects, obviously, pretty much the things that I have talked about, some initiations of studies, some tests, certification projects, as well as a patent project that we have for actually both projects. We feel that there are 3 main reasons to invest in Cline. Cline is today very lowly valued, has a low market cap. We have projects in large market segments with very large market potential. And we have patented technology with approved patents in all the major markets. And we are also continuing that patent strategy to strengthen both projects, as a matter of fact. And that is what I will end up with. Thank you for listening. And now I guess there will be questions.
Thank you, Patrik, for that presentation. I would like to start off with a question about StemCart. And what would you say are the biggest benefits seen from both a patient perspective and a health economics perspective?
Well, from the patient perspective, it's pretty clear, I think. You can treat more people earlier, less invasive. It is going to be invasive, but much less invasive than the surgeries today. And the the rehab times I believe, we don't have proof of that yet, but it's obvious that that's going to be much less. Risk of infection during surgery is not as high here, et cetera, et cetera. So from the patient perspective, there are many advantages. From a healthcare perspective, I think some of those can be translated as well. You can treat more patients. Less rehab is less costly. Less use of resources, surgeons, operation rooms, etc., is also less costly. Not to mention side effects, which you always have when you do surgery. One aspect that somebody brought up is people who are in chronic pain. They also use a lot of pain medication, which eventually is a cost for both the patient as well as the healthcare system. So, depending on which type of healthcare system you have. So that's another aspect that is very often overlooked, I think.
How does the competitive landscape, what does it look like? Are there any more companies doing the same thing that you do, if we're looking at the stem cart still? Yes, there are a number of companies trying to bring out not the same thing that we do, but towards the same illnesses or problems that people have. The problem so far is that no one has been able to produce a cartilage or chondrocytes of similar quality that or identical quality that you have in your body. In other words, quality high enough so that it lasts. And that's a key aspect of these types of cell therapies, because a surgery or a prosthesis that you have only lasts for about 10 years. Then you have to redo it. That's why you try to push surgery as high up in the age group as possible, so you don't have to redo it. And you don't want to have the same thing with this type of therapy. Then you have gained very little. So that is the problem. We believe we have a method for that, and that's will be our big advantage. But are there others trying to do, as you said, similar things? Yes, it's a big area. It's one of the largest area in healthcare, in the healthcare industry today. So yes, there are other competitors. It's a good thing. It's you have competition, but it's a good thing too, because we don't have to drive this ourselves, and it'll be interesting to see what the future has to offer there. So if we move on to the cell race, you have had a collaboration before with Live Diagnostics. You also Correct. Had a license together with them. And could you tell us on the road from the collaboration to the acquisition, and why did you choose to go down that way? Mm-hmm. Well, we started out as a supplier, basically, of small surfaces to that project. We collaborated from the very start with them. They chose our technology to work with. When Live Diagnostics talked to investors, as well as we talked to investors for Cline, many investors commented that they thought it was a better idea to have all the IP under one ownership. And personally, maybe I shouldn't say this when you have investors listening, but personally, I don't understand that. I've worked in the life science industry for 30 years, and most companies have many licenses and deals. Big pharma, for instance, if you go into a factory, they have hundreds of licenses they have to use. So I don't understand that really. But instead of me trying to convince people to do it my way, we decided together with the two main owner groups that why don't we collect this under one umbrella, do it under Cline's umbrella, and to sort of take away that argument. And that's the main reason. And we have collaborated from the very beginning, so we know very well what they're doing, and we've taken over most of the personnel, etc. So it wasn't really a problem. We actually share the cell culture lab at AstraZeneca BioVenture Hub before, so there's very little change apart from the ownership. How have the results from the studies you've made so far in the cell race, can you tell us about the studies you have made, and how does the road looking forward look like? Well, as I presented here, or mentioned, we have gotten to the point where we have finished the studies on cell lines, which you usually do. Those are prepared to act in a certain way.
So what we could say is our prototype works, it's robust, and now we have just moved on to fresh patient material to make sure that that material behaves in the same way. Before you start a big study, you have to do some testing to make sure that you don't have to adjust, that it works. Other aspects, how does the cell preparation need to be taking place? How do you get the material from the pathology lab to, right now, to our lab? In the future, the product is supposed to be at the pathology lab, so that is sort of a special thing. But those things need to be taken care of, and that's what we're doing now. That if everything goes to plan, according to plan, then we're done with that by the end of the year. But as you know, things can sometimes not go to, according to plans, and then we have to change. But right now it looks very good. It's very promising, but we need to take the next step, and the next step is to involve clinics. We have a couple of involved already, but we need to have more clinics to get more material from different parts of the world involved, because because eventually we have to compare it to the methods used today. And as I think most people know, cancer diagnostics are looked upon differently in different parts of the world. As a matter of fact, they're looked differently from our city to your city. So it's very, very personal. So that we need to sort of collect together and pool to make sure that we have a prototype as well as our analysis aligned with what is accepted. And then hopefully next year we can start then a larger study called a clinical relevance study, or the first of them, and take it from there.
We're moving on to some questions from investors.
Mm-hmm.
And the first question is, you want to sell your project or products when they are not completely ready.
Mm-hmm.
How do you convince a potential buyer to buy a product that is not ready?
It is, in a way, a good question, but the answer is twofold. One, you have to have a good product. You have to have robust results. You have to have come far enough, that is, close enough to an approval or whatever is necessary for that product, for them to be interested, and the market for them, the market segment has to be large enough for a big company to be interested. And in our case, we know pretty much where that is. If we have been able, if we do our first clinical relevance study and get good results, that's where we can be in that case. And we know that because we're already talking to a few. You can also answer that question in this way. There is not a single product from a small life science company that is ever ready when it's sold to a larger company. The whole idea is for the larger company to make it ready or take it to the market. So this is a normal procedure in this business. We're not talking about a takeover or an acquisition of a company. We're talking of a product that has promised to with a big company's help, sell for a lot of money globally. And that's why you need a big company. A small company can never attain these big global potentials that we talk about, that every single life science company talks about. So it's a standard procedure in our business, not so much in other types of businesses.
The next question from investor is, how do your sales strategy, what does it look like? Can you tell us about the work?
Well, I have, again, to answer that twofold, because we do sell our small surfaces to researchers, and we will continue to do that, to be able to attract other projects or be able to build it into, or embed, as I said, into other projects. And we have done that quite a bit in the past, not so much now because we're spending our time and efforts on these two projects. But we will continue to do that. As much as we can. As a matter of fact, we already have some other projects potential, but we don't have the resources to make them projects within Cline yet. As far as if you're talking about the strategy to sell our projects to larger companies or attract larger partners, collaborators, if you will, it's also a standard thing. You have to go out and meet with them. You have to take part in meetings, congresses, conferences, etc. And then they have to get to know you. And normally you don't make that type of a deal like this. You know, they need to get to know you, understand what your project is, follow the project and the progress that you make during sometimes many years. Hopefully, in our case, not that many years, but I'm sure they will have to follow us for a year or two before they decide to, in that case, go with our product and be interested in it. So that's what we're doing. We've already started that, but we will accelerate that as we get more results and more things to show.
And the last question from an investor is what milestones would the rights issue that you're now doing, what milestones do you see achieving with that money?
Well, again, back to the presentation, it's in there. For the cell race project, we will reach a stage where we can start this, as I call it, larger or a first clinical relevance study. We can't complete it, but we can start it, and that's the milestone. For the stem card project, we will at least have a phase 1 clinical phase 1 study planned. Doubtful if we can start it, not necessarily because of the money, although money is a factor in clinical phases, but I don't think we have time to get to that stage. But it should at least be planned, and there are some steps that I showed up on in the presentation that we have to take in order to get there. We need an EMA certification, etc., etc.
Thank you for coming here today, Patrik. We look forward to following you.
Thank you very much.
Thank you.